Welcome to all. Myself Sajithra, lecturer of Emmanuel Arasar College of Pharmacy. In this section, I discuss about electron chain transport. Electron chain transport is the transfer of electrons from NADH and FADH2 to oxygen via multiple carriers. The electrons are derived from NADH and FADH2 combined with oxygen, and the energy released from these oxidation or reduction reaction is used to derive the synthesis of ATP from ADP. This transfer of electron is done by multiple carriers which constitute the electron transport chain. Mitochondria is the site of electron transport chain, and this is the structure of mitochondria. Components of electron transport chain. First, complex number 1. Complex number 1 is named as NADH dehydrogenase, and it has totally 46 proteins, and it has FNM and 9 FES centers. Complex 2, it is named as succinate coenzyme Q reductase. It has 5 proteins and it contains FAD, cytochrome B, 3 molecules of FES. And third complex is named as coenzyme cytochrome C reductase. It contains 11 proteins and it contains cytochrome B, cytochrome C1, iron and sulfur molecules. And complex number 4, it is named as cytochrome oxidase and it contains totally 13 proteins. Complex number 1, which is NADH dehydrogenase. Complex 1 catalyzes oxidation of NADH with the reduction of coenzyme Q. It includes at least 46 proteins along with prosynthetic group FMN and several iron-containing sulfur centers. It pumps totally 4 protons across the mitochondrial membrane. Coenzyme Q, it is also known as cubiquinone. It is a benzoquinone linked to a number of isoprene group units. Coenzyme Q is very hydrophobic. It dissolves in the hydrocarbon core of membrane. It, it happens in 3 redox states that are fully ionized, partially ionized, and fully reduced. Only electron carrier that is not a protein-bound prosynthetic group. Complex 2, which is known as succinate dehydrogenase. Succinate dehydrogenase of the Krebs cycle is also called complex 2 or succinate coenzyme Q reductase. It is the inner mitochondrial membrane bound proteins, and FAD is the initial electron acceptor. FAD is reduced to FADH2 during oxidation of succinate to fumarate. FADH2 is then reoxidized by transfer of electrons through a series of 3 iron sulfur centers to coenzyme Q yielding QH2. It does not pump any proton during the transport of electron across the inner mitochondrial membrane. X-ray crystallography analysis of E. coli complex 2 indicates a linear arrangement of electron carriers within complex 2, consists with the pre-direct sequence of electron transfers. Complex 3, coenzyme Q cytochrome reductase. Complex 3 accepts electron from coenzyme QH2 that is generated by electron transfer in complex 1 and 2, and it releases 2 protons into the transmembrane space. Within complex 3, the released electrons are transferred to an iron sulfur centers and then to B-type cytochrome or cytochrome C1. Finally, the 2 electrons are transferred to 2 molecules of the oxidized form of cytochrome C. 2 additional protons are translocated from mitochondrial matrix across the intermembrane space. This transfer of proton involves the protein involves the proton motive Q cycle. Cytochromes. Cytochromes are protein with the heme prosthetic group. They absorb light at characteristic wavelength. It carries electron 1 at stage time to complex 4. Major respiratory cytochrome B, C, or A in electron transport chain to a type of cytochrome, that is cytochrome A and cytochrome A3, and then it contains two B-type cytochrome, that is cytochrome B1 and cytochrome B2, and it contains two C-type cytochrome, that is cytochrome C and cytochrome C1. Cytochrome C is a key regulator of a programmed cell death in mammalian cells. Complex 4, cytochrome oxidase. It catalyzes the transfer of electron from reduced cytochrome C to molecular oxygen. It contains 13 subunit, 2 heme group, that is heme A and heme A3. 3 copper ions arranged as 2 copper centers designed as a copper A and copper B. Copper A contains 2 copper ions linked by 2 bridging disulfide residues. Copper B is coordinated by 3 histidine residues. 2 protons per pair of electrons are pumped across the membrane and another 2 protons are transferred to molecular oxygen to form water. Metal centers of cytochrome oxidase, that is complex 4, heme A and heme A3, and copper has two forms, that is copper A and copper B. O2 reacts at a, that is oxygen react at a binuclear center consisting of heme A3 and copper B. Electron enter complex 1 at a time from cytochrome C to copper A. Then they pass via cytochrome A to the binuclear center where the chemical reaction takes place. Complex 5, that is ATP synthase. Mitochondrial ATP synthase consists of two multi-subunit components, that is F0 and F1, which are linked by a slender stalk. F0 is electrically derived motor that spans the lipid bilayer, forming a channel through which proton can cross the membrane. F0 provides channels for protons. F1 harvests the free energy derived from proton movement down the electrochemical gradient by catalyzing the synthesis of ATP. F1 phosphorylates ADP to ATP. This is the ATP synthase. Chemoosmotic hypothesis. This hypothesis couples electron transport to ATP generation and that ATP is generated by use of energy stored in the form of proton gradient across biological membrane rather than by direct chemical transfer of high energy group. Complex 1 and complex 4 appears as a proton pump which transport proton across the membrane due to conformational change induced by the electron transfer. In complex 3, protons are carried across the membrane by ubiquinone. Complex 1 and 3 form 4 proton per pair of electrons. Complex 4 pumps 2 proton per pair of electrons transported and other 2 protons are combined with oxygen to form water. And it's the summary of electron transport chain. Totally, on this one circulation of ATP, totally 10 protons, that is in one NADH synthesize 2.5 ATP and one FADH2 synthesize 1.5 ATP. And total ATP consumed is from pyruvate dehydrogenase NAD, that is it has NADH, it consumes 2.5 ATP, and then Krebs cycle, it totally have 12.5 ATPs. What about NADH from glycolysis? NADH is made in cytosol, cannot get into matrix of mitochondria. It involves two mechanisms. First one, muscle and brain, and second one, liver and heart. And inhibitors of electron transport chain is um, rotenone is a drug which inhibits the complex 1, and amytal is a drug which inhibits the complex 1, and um, antimycin A which inhibits complex 3, and cyanide, acide, carbon monoxide which bind to the complex 4 and inhibit the transfer of electron to oxygen. Uncouplers of electron transport chain 2,4 dinitrophenol, dicumarol, carbonyl cyanide, fluoromethoxyphenyl hydrazone. Thank you for watching.